going to get started. Although a few people are joining now. Gosh, we went from 2 to 7, so that's pretty good. Let's close that. And I don't think I need that any further. Oops. Let's not close that. So we're supposed to be starting chapter 14, Principles of Disease, today. Let me remind you that there will be a lab on Friday. Excuse me, not a lab, a quiz on Friday. It's over chapter 8 and lab 4. I haven't finalized it, so you won't find it yet, but I will try and get that done after class today. Any questions about any of that? All right, so I showed you this last slide of chapter 13, correct? Anyone want to comment, or Christopher, if you can nod your head? Okay, thank you. So let's begin with chapter 14. Please also read the unknown project instructions file. I'm going to be talking about that next week. So chapter 14, Principles of Disease and Epidemiology. Looks like there's a mistake there. There should be a space in there. The major goals and rough outline is given here. Know the terms pathology, etiology, microbial antagonism. Understand the relationship of the normal microbiota and its host. And let me back up a little bit. On this chapter, about 50% of it is understanding and knowing the terms, because there's a large number of terms in this chapter, and a lot of this chapter is understanding the terms. Any questions about that? All right, we'll talk about the stages of disease development, that most diseases go through the same stages of disease development, meaning they start the same and then they end up the same. And then lastly, understand the nosocomial infections and how to control them. Any questions about any of that? So the first terms to know are pathology, the study of disease; etiology, the study of the cause of disease. For example, the etiological agent of anthrax is Bacillus anthracis. Pathogenesis, the development of a disease. And so those are 3 terms you need to know right off the back when we're studying the principles of disease and epidemiology. Know that an infection is the colonization of the body by pathogens, or the presence of microbes where they're not normally found. So when we say someone has an infection, they're colonized by human pathogens, or they have microorganisms growing where they're not normally found. Disease is defined as an abnormal state in which the body is not functioning normally. Any question about any of those terms? Once again, understand that few microorganisms are pathogenic, and if they are pathogens, it means that they are found where we're talking about the normal microbiota. If it is causing disease, then it is because it's growing where it should not normally be. So few microorganisms are pathogenic, and some microorganisms actually benefit the host. Generally speaking, the normal microbiota benefit the host. Any question about that? So let's talk a little bit more about the normal microbiota and the host. We have talked about it before, but we're going to dive into it here deeper. The normal microbiota is a large community of microbes that are found in association with all animals and plants. And generally with animals, they don't come into the animal until after they're born. These communities are known as the normal microbiota, microbes that permanently colonize a host. So they get on or in the host, and then they permanently will be a part of that host's normal microbiota. They are found only in certain locations, meaning the microbes are found only in certain locations. Obviously the skin and the mucous membranes and all of the internal mucous membranes. So the normal flora is found in the gastrointestinal tract, and it's found in the upper respiratory tract, the lower urethra. It's actually found, we'll talk a little bit about that later, but it's found a little bit higher than just the lower urethra. They've discovered, at least in women, I don't know if they've discovered that in men or not, the studies I've read have all been in women, that the bladder actually has a normal microbiota at low numbers, but they have a normal microbiota, at least in the female bladder. The normal microbiota is only found in certain locations, and we have a disruption of the normal microbiota and the host balance. And when either the normal microbiota is thrown out of balance or the host is thrown out of balance, it can change the the relationship between the normal microbiota and the host. And it appears to be associated with disease when that balance is thrown off. It can also be associated with impaired immune function. So if the normal microbiota is thrown out of whack or out of balance, then the host can get disease or impaired immune function. Any question about any of that? So where is the normal microbiota normally found? Well, obviously, all of the external coverings of the body, the skin, the mucous membranes, the internal mucous membranes, which most of that's not shown, but the throat, the stomach, the intestines, and then the upper respiratory system. Usually the lungs do not have microbes in it. And if there are microbes in the lungs, that is a disease. Any questions about that? Microbes are not normally found in the internal blood, cerebrospinal fluid, muscles, nervous tissue, bone, anything internal to the body. The normal microbiota is not found there. Now that's the normal microbiota, which colonizes the host essentially permanently, meaning those species will be with the host throughout the host's lifetime. That's different from a transient microbiota, which may be present on the host for days, weeks, or months, but unlike the normal microbiota, does not permanently colonize the host. An example of that was my aunt had a Staphylococcus aureus infection, and we went to see her, and when I showed up, she gave me this really big hug, and of course she did that also when I left. And after hugging her in the lab, I then tested myself. I just routinely did it back in those days. In the lab, I tested myself for the presence of whether or not I had Staphylococcus aureus. And after hugging her, I actually had Staphylococcus aureus on my skin. And I said, oh geez, I must have picked this up from my aunt. And Staphylococcus aureus, if you don't know, is a human pathogen, but it is found on the skin of about 20% of Americans. It's found in the nasal mucosa, and where else? Maybe the throat of about 50% of Americans. And I did occasionally test my nose, but I didn't really do that very often. But I tested my skin about every 3 months. And after I hugged her, I had it. And I was a little bit worried about that, and I said, well, just have to keep an eye on it. And then the next time I tested my skin, which was several months later, it was about 3 months, I don't remember, it was gone. And then I tested it again 3 months later, and I never saw it ever again. So that was a transient microbiota, okay, that I probably picked up from my aunt who had a skin infection, and I should have tried not to hug her when she had that. Any questions about any of that? So let's talk about the normal microbiota. When we talk about a human, an average human has about 1 × 10 to the 13th human cells in their body. Okay? And the average human has about 1 × 10 to the 14th bacteria cells as part of their normal microbiota. That means they have 10 times more bacterial cells than they have human cells. And that asks the question, are we more microbe than we are human? Okay? Any questions about that? Don't let that freak you out if you get freaked out about that. Showing you some pictures of the normal microbiota, a large number of bacteria, the orange spheres on the surface of the nasal epithelium, the normal microbiota in the stomach, and then bacteria in the large intestine. You can see both cocci and bacilli there. The human normal microbiota has been found in some surprising places recently that people didn't think had the normal microbiota. That they were finding they were finding bacteria in urine, and somebody went and investigated and found that at least the female bladder does have a normal microbiota. It's not at high numbers, and it's only certain species, but the bladder actually has, at least the female bladder, has a normal microbiota. And then the human breast tissue, we're talking about females, does have a normal microbiota in the breast tissue. And mother's milk is not sterile. It has a normal microbiota in it. So infants, when they're breastfeeding, they're getting a normal microbiota from their mother. Okay, besides being on the outside of the breast, which the baby will pick up, but the milk itself has normal microorganisms in it. And then the placenta has been shown to have a normal microbiota. Any questions about that? If you don't know, a placenta is what attaches the baby to the mother. So there's a relationship between the normal microbiota and the host. And this is generally a good relationship because the normal microbiota can engage in antagonism, and that's competition with human pathogens. So the normal microbiota can prevent the growth of pathogens by occupying niches that the pathogen might occupy. And so if the skin is totally covered with the normal microbiota and you get one pathogen, that pathogen is likely not going to get established because the skin is already occupied. So they're competing for space. They compete for resources. If the normal microbiota uses up all of the resources in the region, then a human pathogen gets there, it can't grow. And if it doesn't grow, it's probably not going to colonize the host. And it is well known that you generally, with bacteria, you need more than one cell to become infected with something that causes infection with a low number of cells, something like salmonella. If you just get one salmonella cell in your gut, it's likely not going to colonize your gut because the normal microbiota are going to be antagonistic with it, and it won't grow and colonize your gut. On average, humans need to consume about 500 salmonella before they will colonize you and cause disease. Any question about any of that? And that's normally from your normal microbiota competing for space and resources. They can also compete for or cause antagonism by other ways, like the normal microbiota on the skin and in the urethra and in the vagina. It makes acid, and it makes it too acidic for most human pathogens to live. So the production of acid can prevent human pathogens from colonizing humans. And then some normal microbiota produce bacteriocins, which are like an antibiotic, but except they're much more narrow in the species they affect. Like, for example, E. coli that produces bacteriocin, it's a protein that acts like an antibiotic. But instead of knocking down all microbes, the bacteriocin from E. coli only knocks down related bacteria, such as Salmonella and Shigella. Any questions about that? And I always wondered why somebody wasn't using bacteriocins to be working like an antibiotic, because if somebody got a Salmonella infection, a bacteriocin from E. coli would work. And I read a few years ago that people are experimenting with using bacteriocins now for an antibiotic. I haven't heard anything further, but at least they're being experimented with. So the bacteriocin from E. coli only knocks down Salmonella and Shigella. It wouldn't knock down, what's another species? Like a Bacillus species of bacteria. I was trying to think of a species of bacteria. I can't remember. Something that we have in our gut, and I'm not remembering another name. Obviously E. coli, but I was trying to think of another bacilli we have in our gut, and it doesn't knock down that species. Whereas if you used an antibiotic, it would knock all types of bacteria given a certain type. Like if you use an antibiotic that hits gram-negatives, it would hit all gram-negatives. A bacteriocin wouldn't. And then the relationship with the normal microbiota and its host has led to the development of probiotics, where people consume live microbes for their benefit, meaning they consume these microbes to help colonize them and then get some benefit for that. Like women who eat yogurt, they're less likely to get a yeast infection. And it's because of the microorganisms in the yogurt competing with the yeast and making a yeast infection less likely. Any question about any of that? So the relationship between the normal microbiota and the host is a relationship of symbiosis, where two organisms or two populations live together. Now the normal microbiota has two symbiotic relationships. The species can be following commensalism, or they can follow mutualism. If the normal microbiota is commensalistic, that means one organism is benefiting and the other is unaffected. I'm trying to think of an organism like that would be, and it's kind of hard to think of one that gives no benefit to us. My phone is ringing. Oh well, we'll answer it later. A microorganism on the skin that doesn't give us any benefit, maybe Micrococcus luteus. It benefits because it's finding a home with us, but we might not get much benefit out of that relationship, okay? That would be different than a mutualistic relationship where both organisms benefit. Like Staphylococcus epidermidis benefits because it gets a home, it lives on our skin. And then we benefit because Staphylococcus epidermidis makes the skin more acidic and will compete with other pathogens on the skin, such as Staphylococcus aureus. So that is a mutualistic benefit, or mutualistic relationship. Any question about that? Now, a human disease-causing organism, a pathogen that causes disease, and if it colonizes the host, that would be a parasitic relationship, where one organism benefits, it's given a home, and it has nutrients that we supply to them, and then it harms us. And that would be when you have a skin infection that's caused by Staphylococcus aureus, or a C. diff infection inside your gut. That would be a parasitic relationship. And the human pathogens follow a parasitic relationship. Any question about any of that? So the normal microbiota do not normally cause disease, but they can be opportunistic. An opportunistic microorganism is one of the normal microbiota that does not normally cause disease, at least in healthy individuals, but under certain circumstances they can cause disease. Like if they colonize an area where they're not normally found. Like normally I have Staphylococcus epidermidis on my skin, and if it's growing on my skin, that's fine. But if it causes disease inside my skin, in the tissue of the flesh, of the skin, that would be an opportunistic microorganism, because it doesn't normally grow there. But if I had a puncture wound or a cut, it could invade and cause an opportunistic infection. Normal microbiota do not normally cause disease, but what happens in an immunocompromised person? Sometimes the normal microbiota can cause disease and become an opportunistic pathogen. And that usually happens because the immune system isn't functioning or not functioning well, and then the normal microbiota starts growing in regions where they should not normally be, or in areas where they normally are, but they are growing in much greater numbers than they should be. Like all of us have a few cells of Candida. Oh, gosh. gosh, what the heck is that? Candida albicans on our skin, and it's part of our normal microbiota. And that's fine. But if you take an antibiotic that can hit your bacteria on your skin, and then it's not going to compete, the bacteria, with the yeast, and then you can get a yeast infection on your skin because the yeast isn't competing now with the bacteria, and you get a bloom of yeast growing on your skin, causing a yeast infection. All right, so that's some opportunistic pathogens. Another one would be E. coli. It's normally found in the gut, and if it's growing somewhere else, that would be an opportunistic pathogen. And I mentioned Staphylococcus aureus is found in about 50% of Americans in the nose and in the throat. But if it's growing on the skin, it can cause skin disease. Any question about that? All right, if there's no questions, let's change and move on to the etiology of infectious diseases. Dr. Koch postulated how to show that a given organism causes a given disease. Let's pull that up. And he published this, and they're called Koch's postulates. What we use to prove that a given organism causes a given disease. And Dr. Koch showed this with Bacillus anthracis, showing that it causes the disease anthrax. When you have a sick organism and you take microbes out of that organism and then grow them up in the lab and isolate them and obtain pure cultures, and if you find one pure culture that when you inject into the organism and makes the organism sick, why the bunny is turned on its side here, it's sick now, poor bunny. And then that's still not enough. Dr. Koch says you now have to isolate from this sick organism the same organism that you isolated from the first sick organism. And if all of these are in place, and this organism is present in every case of the sick organism, then you show that this organism causes this disease. And that's what Koch's postulate is. Any question about any of that? Those are the steps we use to show that a given organism, like Bacillus anthrax, causes a given disease, such as anthrax. Now, there are exceptions to Koch's postulates. What do you do if you can't grow up the culture in the lab? Obviously, we can't use Koch's postulates then, because the organism won't grow up in the lab. We just don't know how to grow it. And there are some examples of that, like Bartonella henselae, the disease that causes cat scratch fever. We just can't grow it up in the lab. We can grow it on a cat, we can grow it in people, but we can't grow it in the lab. So we can't use Koch's postulates on that. And what do you do if multiple organisms cause the same disease? So you see multiple organisms causing in people the same disease signs and symptoms. Then you can't use Koch's postulates. As you recall, we mentioned that there are 3 families of viruses that cause the human cold. So we can't use Koch's postulates to say that one species causes the human colds. And what do you do if one species causes different disease conditions? You can't use Koch's postulates because The same species causes different signs and symptoms. Can anyone think of an example of that? I have one, but it's not real good, and that is salmonella can cause two different diseases. It can cause salmonellosis, and it can cause typhoid fever. Typhoid fever is much worse than salmonellosis. The patient is much sicker and the disease lasts a lot longer. The trouble is, typhoid fever is really just a really bad case of salmonellosis, meaning the signs and symptoms are similar. It's just that typhoid fever is worse. Any question about that? But there are some diseases, I just can't think of an example, where one species caused two different disease conditions. Maybe we could state with COVID-19, sometimes people get flu-like symptoms and a fever with COVID-19, and then other patients, they get either diarrhea or constipation, and that might be the only symptom you get with COVID-19. And then what do you do if there's moral and ethical reasons for a human-only disease? Can you use Koch's postulates? No, you can't. Like HIV, we've never injected it into people to see if it causes AIDS, because AIDS would be such a horrible disease, you don't ever inject that into humans. Okay? So for moral and ethical reasons, we can't use Koch's postulates to show that HIV causes AIDS. So how did we go about finding that HIV causes AIDS if we can't use Koch's postulates? Anybody have any idea? Any guess? Nobody's going to take a guess? I'll give you an extra credit point.
Did you take samples from their blood?
Yeah, Ian, if you take large epidemiological studies and then just followed people over time, some of them became infected with HIV over time, and then you follow those patients, and some of them later, eventually all of them, later came down with AIDS. And for following those large epidemiological studies over time, we were able to show that HIV causes AIDS. Ian, just email me and I'll get you the extra credit point. Okay, any questions about any of that? If not, let's go on to move on to the topic of classifying infectious diseases. There's a lot of terms for classifying infectious diseases. When we say a patient has a symptom, that means they have a change in their body function that is experienced by the person from a result of disease, but only that person can experience it. Nobody else can measure it. Does that make sense? I'll give you an example. When somebody has a headache, only that patient knows that they have a headache. There's no way to measure a headache. Okay? Nobody else can experience, nobody else can measure it. So it's subjective. A symptom is one that the patient experiences, and only they can experience it or measure it. A sign is a change in the body that can be measured or observed from an outside person. Like when a patient has a temperature, that can be measured. Any question about that? Now you'll find with patients that sign and symptoms, they're sort of used interchangeably in the general public. And generally what I do is I'll just say sign or symptom or sign and symptom, meaning I don't try to distinguish between the two. But by the textbook, a sign is one that you can measure. And I suppose it's important because we can verify that one. A symptom, you just have to go on what the patient says. A syndrome is a specific group of signs and symptoms that's characteristic of a disease. So you could talk about the signs and symptoms or the syndrome of AIDS. Okay? Any question about that? A communicable disease is any disease that is spread from one host to another. So this is a disease that spreads. It's caused by a microorganism if it's a communicable disease. And generally, all diseases that spread are caused by a microorganism. Examples of a communicable disease are measles, TB or tuberculosis, chickenpox. A contagious disease is a disease that is easily spread from one host to another. Like the flu would be a contagious disease. Measles are also a contagious disease. Tuberculosis is communicable but not contagious, meaning it does spread from one host to another, but it doesn't spread easily. And so all contagious diseases are communicable, but not all communicable diseases are contagious. And if you want to think of some that are communicable but not contagious, tuberculosis is an example, and leprosy is another example. You usually need long-term exposure to come down with tuberculosis or leprosy. So it doesn't spread easily. Whereas measles and the flu, you just need short-term exposure and you can get it. Any question about that? Communicable, contagious? A non-communicable disease is one that does not spread from one host to another. That can be, and usually is, well, it can be caused by microorganisms like tetanus. And somebody brought up botulism. When one patient gets it, it's not going to spread from that patient to another patient. Okay? There is one possible exception, and that is when we're talking about cannibals. A cannibal, if he eats somebody who has a non-communicable disease, may get that communicable disease, but cannibalism is so rare among humans that we're not going to discuss it. And tetanus and botulism normally do not spread from patient to patient. Okay? Any question about that? And then another non-communicable disease would be diseases that are inherited, like inherited diabetes. It's non-communicable. Any question about that? So let's move on to talking about the occurrence of a disease. To understand the full scope of disease, we need to know the details about its occurrence, how many old cases there are, and how many new cases there are. The incidence is the fraction of a population that contracts a disease during a specific time. So the incidence is looking at the new cases only. And you can look up that they have an incidence of the rate of new cases of COVID-19 at the moment. And in Arizona, the incidence of COVID-19 has been going up almost exponentially. So there are more and more new cases. Okay? The prevalence is the fraction of a population having a specific disease at a given time. So that means the prevalence is counting the old cases as well as the new cases. An example of that, in AIDS in USA in 2007, the incidence was 56,300 cases in the United States in 2007. The prevalence, though, was 1,185,000 cases. So the prevalence is larger than the incidence, because the incidence is only counting the new cases, those people who recently became infected. Any question about that? When you're counting the prevalence, like patients who had COVID-19, that's also counting people who had the disease and got better. They recovered. Sporadic disease is a disease that occurs only occasionally in the population. Like typhoid fever in the USA happens sporadically. Not very often, but every once in a while it happens, and will happen in one state and happen in another state a few days later or something like that. So it's not happening all the time. An endemic disease is one that is constantly present in the population. Like the common cold is endemic in the United States. We always get it. It is more common in the wintertime than in the summer, although you can get it in the summertime, but it's much less likely. Malaria is endemic to the tropics. If you're traveling to the tropics, then you should take precautions to prevent from getting malaria. Okay? Now, when you're talking about a few cases of disease, generally we say it's sporadic. But when it's few cases, it's hard to say, is it endemic in the United States or is it sporadic? Like some people, and they argue about this all the time, they'll talk about Mycobacterium pneumonia. Is that a sporadic disease in the United States because it happens every once in a while, especially among the homeless population? Or is it actually endemic in the United States? And like I said, they argue about that. And when you're talking about a few cases, it's hard to make a distinction between sporadic and endemic. And we're not going to get into that argument, okay? Any questions about that? Epidemic is when a disease is acquired by many hosts in a given region or on a continent. Like when there would be an influenza epidemic in the United States or in North America, we'd call that an epidemic. Once it spreads beyond one continent, we then call it pandemic. And every pandemic so far has become worldwide. Like when first H1N1 happened, it was actually detected first in the United States, but we then learned that it actually started first in Mexico, and then American travelers brought it back to the United States, and we discovered H1N1 first in the United States, and so it was an epidemic in North America. Well, it then spread and became pandemic, meaning it spread to more than one continent. COVID-19 was first epidemic in China, and we'd say in Asia, and then it spread and became worldwide and became pandemic. Usually, when a disease spreads to more than one continent, it becomes worldwide. Any question about that? We can talk about disease and how long they last and the severity of it. And there we talk about its duration. And that's another useful way for defining the scope of disease in terms of its severity and duration. An acute disease is a disease that develops rapidly, is short-lived, and then it goes away. Can anyone give an example of an acute disease? Symptoms develop rapidly, short-lived, and goes away.
The flu.
The flu would be an acute disease, but a better example is the human cold. Okay? Somebody has the human cold, you get it a few days later, it develops rapidly, you get a runny nose and maybe a sore throat. But then it's short-lived, and usually a human cold goes away within a week or even less time. So it's even better than the flu, because the flu can sometimes last for at least a month, okay? But the cold is short-lived. So that's a good, perfect example of an acute disease. Now, there are many examples of short-lived diseases. Usually chickenpox in a child is an acute disease. The symptoms develop rapidly, and it's short-lived, and then about a week after they first get symptoms, the symptoms go away. Okay? Usually that happens in the childhood diseases where they're acute diseases. A chronic disease is a disease that develops slowly and persists for a long period of time, and usually the disease gets worse over time. A case of a chronic disease is mononucleosis. It develops slowly, gets worse, and it persists for a long time. That disease can last for months, where the patient just is tired and worn out and down. Hepatitis B causing liver cirrhosis would be another case of a chronic disease, where it develops slowly, persists for long periods of time, and gets worse for a long period of time. With many of them, like mononucleosis, you do get better. Sometimes in the chronic disease, the patient just gets worse and worse and worse until they eventually die. There is something in between an acute disease and a chronic disease. I'll mention it, but I'm going to say I'm not going to ask you about it. We call it a subacute disease, which has symptoms between an acute disease and a chronic disease. And if you've ever heard of subacute sclerosing panencephalitis, that would be a subacute disease. But I'm not going to ask you to know that one, okay? The extent of the host involvement is another thing to look at with disease. Infections can be classified according to the extent to which the host body is affected. A local infection is when a pathogen is limited to a small area of the body. Like on the skin, a local skin infection can cause a boil or an abscess, and that would be a local infection. If it's a boil, it might be caused by Staphylococcus aureus, but it's localized, only in that one area of the skin. A systemic infection, also called a generalized infection, is an infection throughout the body. Like when the patient comes down with a measles infection, it becomes systemic. The measles spreads throughout the body. Any question about that? If Staphylococcus aureus grows out of the boil, gets into the blood, the Staphylococcus aureus infection can become systemic, in which case you could have Staphylococcus aureus growing in the blood and throughout the body, because the blood will carry it throughout the body. A focal infection is a systemic infection that began as a local infection. So the case of Staphylococcus aureus starting as a boil and then spreading and infecting the blood, and then the blood carrying it to the rest of the body would be a focal infection. So that would be a focal infection. Another one is MRSA, which is really the same thing. MRSA is multi-drug resistant Staphylococcus aureus, and if it spreads from the skin to the blood, it would be a focal infection. We talked about bacteria getting into the blood. There were two terms to cover that. Bacteremia is the presence of bacteria in the blood. Septicemia is the growth of bacteria in the blood. That's slightly different. In both cases, you have bacteremia, but if the bacteria are actually growing in the blood, then it's septicemia. Usually when a patient has bacteria in the body, it's also septicemia, meaning the bacteria are growing in the blood. I can't think of any cases where that would not be the case, but I imagine there are some bacteria that just find the blood unsuitable to grow in, but the blood carries the bacteria to other places in the body, and then it can colonize other places and grow in that place. I can't think of an example of that, but it's possible. Toxemia is the presence of toxins in the blood. We haven't talked much about toxins yet, but some bacteria can secrete something from their cells which are toxic to us, and that we call a toxin. Any question about that?
Are you talking about like when a gram-negative cell, because of their little lipidase, release their toxins?
That could be the case. I wasn't going to talk about that because we're talking here about the presence of toxins in the blood. On a gram-negative cell, the toxin is not released until the cell dies. So if a patient had a blood infection, meaning they had bacteria in the blood, and then they were given antibiotics which would kill the cells, then we would see toxemia, toxins in the blood. Okay? All right. Viremia is just the presence of viruses in the blood. So talking a little bit more about the extent of host involvement, the state of the host resistance also determines the extent of an infection, meaning how well the host immune system is, how good the host health is, can determine how the host responds to the disease. Meaning someone who their health isn't real well and they get an infection, they're more likely to get a severe infection as opposed to someone who's healthy and they get the same infection. They're much less likely to have a severe infection. A primary infection is an acute infection that causes the initial illness. So when you have a primary infection, that's where the bacteria or the virus first gains entrance into the body. Like if you have Staphylococcus aureus growing on your skin, that would be the primary infection. A secondary infection is when an opportunistic organism infects the body after a primary infection has weakened the host's immune system or weakened the host's defenses. And so an opportunistic infection would not happen, the secondary infection, until after there was a primary infection, which weakened the host and then allowed an opportunistic infection, what we call a secondary infection. Any question about that? Sometime with children, they get sick and they get a sore throat, and then later they get a secondary infection of their ears. The primary infection allowed the secondary organism to get established and cause another problem. I can't think of another example of that, but that's one. A subclinical disease is the same thing as an asymptomatic disease when a patient has no noticeable signs or symptoms. For example, some people carry poliovirus, but they never get signs or symptoms of polio. And as you know, in COVID-19, some people get subclinical or asymptomatic disease, where they're carrying around COVID-19 and they can infect other people, but they have no signs or symptoms of disease. Any question about that? So when we're talking about severity or duration of the disease, it's important to discuss what is a latent disease and compare that to an asymptomatic disease or a subclinical disease. Asymptomatic disease or subclinical, the patient has no signs or symptoms. A latent disease is where the disease first hits the patient, and then the patient goes latent, where they have no signs or symptoms. The pathogen is inactive while the patient is in the latent disease. And then later, the patient comes out with disease again, and the disease comes out of latency, causing disease. The best example of that would be when a child gets chicken pox, and then the agent goes latent. It's staying in the patient, and then many years later, maybe many decades later, the agent comes out of latency and then causes shingles. Okay? In the patient, while the patient is in the latent disease, the patient has no signs or symptoms, and actually the patient has no disease and cannot be infective in the latent disease. Okay, so while the virus is in latency, the patient is not contagious, is not communicable, I should say. Any question about that? Most latent diseases are caused by a virus. I can't think of one. Well, maybe Mycobacterium tuberculosis can go into latency. And then can, especially when the patient's immune system weakens, come out of latency. Yeah, there are a few, but most latent diseases are caused by a virus. And a lot of them are caused by herpes viruses, like cold sores is a latent disease. Herd immunity is when many immune people are in a population, and then that protects the rest of the population from coming down with disease. So if a lot of people are immunized, the people that are in yellow here, then the disease doesn't spread much, because when it spreads to an immune person, the disease doesn't get hold, and so the transmission of the disease doesn't happen. And that means the non-immune people are protected because of herd immunity, that the immune people are protecting the non-immune people. And this herd immunity has a different threshold for each disease. With COVID-19, we will need 80 to 85% of the population immunized before COVID-19 will stop spreading. Okay? And what happens is it can spread if you come into contact with, and you're not immunized and you come in contact with a sick individual. But because of enough people who are immunized, it doesn't spread beyond that. So this individual is protected because he doesn't or she doesn't come into contact with someone who actively has disease. Now with the measles, we need 95% of the herd immunized. Otherwise, the virus does spread. So measles is very contagious, and you need the herd immunity, or meaning you need the immunized people to be very high before the disease doesn't spread. And that's a problem, and that is parents don't really like to vaccinate their children. They stop vaccinating them, and they say, measles isn't a problem anymore. And when the population gets less than 95% immunized, measles does spread. And so every once in a while we have an outbreak of measles where it starts spreading among non-immunized children. Any question about that? So if COVID-19 at the moment would be this type where there's no immunization, and so when somebody gets it, everybody in contact with them will get it, and then everybody in contact with them will get it, and then everybody in contact with them will get it, and then eventually most of the population would get the disease. And that's why you need to prevent your getting the disease until they come up with a vaccine. And you're going to have to take steps to prevent yourself from getting the disease at least until the winter. It's unlikely that there will be a vaccine before the winter. But they are working on a vaccine, so just stay with it. You have, 8 or 9 more months before there will probably be a vaccine. All right, any question about herd immunity? Let's talk about the patterns of disease. Normally, a specific sequence of events is associated with a given microbe causing a disease. So normally, the microorganisms must come from a reservoir, and that reservoir must exist for where the disease organism is coming from before it comes to a susceptible host. So a reservoir is just where the disease comes from. We'll talk about different reservoirs in a minute. And then the pathogen must be transmitted to a susceptible host. Something has to carry it from its reservoir to the host. And it might be the patient is susceptible and it just comes to the reservoir. Okay? But it could be like a fly carrying it from the reservoir to the host. And we'll talk about different things that can transmit disease. And then the pathogen has to invade the host and multiply within the host before it can cause disease. And then besides this, because actually an asymptomatic patient can follow all of this, the pathogen then has to injure the host before we get illness. If there's no injury to the host, or the injury is very, very minor, then the patient will be asymptomatic. And then the illness ends in one of two ways. The host immune system could either destroy the pathogen and then the patient gets better, or in the other case, the patient could die from the pathogen. Any question about that? So that pattern of disease development happens essentially with every disease. Like HIV, it used to be, most patients, they would end up dying. That's actually not the case anymore. They have enough medications, excuse me, they have enough medications that patients are living long term. And there's even been a few patients who they've said it looks like the patient has beaten HIV, and they looks like the immune system rid the body of HIV. I'm not sure how they can do that, because if they have HIV in their genome, how are you going to know that until many years later and it comes out again? But it looks like there's some patients who look like they've gotten better and they no longer have HIV, at least growing in them. And then many cases, like when you get the cold very rarely ever kills a patient. It looks like the immune system destroys the pathogen and then the body gets better. There are some predisposing factors that can make the body more susceptible to, and therefore more likely to come down with the occurrence of a disease. For example, females have a shorter urethra than males, so they have a higher incidence of a bladder infection, a urinary tract infection. Okay? Any question about that? There are inherited traits, a sickle cell gene or sickle cell anemia, that makes the patient more likely to come down with disease. Climate and weather can affect the occurrence of disease. I think we all know that in the wintertime, you're more likely to come down with the flu or a cold in the wintertime than you are in the summer. Fatigue can increase the chance of you coming down with disease. College students are frequently fatigued, and so they're more likely to come down with a disease than non-college students. Any question about that? Age predisposes a factor, is a predisposing factor in coming down with a disease. Normally, for most diseases, the very young and the very old are more likely to come down with disease and are more likely to be hit severely with that disease. That's normally what we see. With COVID-19, we're not seeing too much of that. It's only the elderly that are coming down with disease and the severe cases of the disease. You have to go to very, very young. It is true that newborns can have severe cases of disease. And there's a lot of newborns that come down with COVID-19 that have to be hospitalized. But even among the newborns, there aren't too many of them that die. Okay? So COVID-19 is unusual in that, like I said, usually with disease, it hits the young and it hits the elderly. Lifestyle can also make you more susceptible to disease. For example, people who smoke, they're more likely to come down with a respiratory infection. Chemotherapy can also affect whether you get disease. People who take a chemotherapeutic drug, that usually weakens their immune system, and so they are more likely to come down with disease. Any question about any of those predisposing factors? And remember, you have a predisposing factor, so you have to protect yourself from getting disease. Any questions? The development of disease typically follows the same development pattern. Once the host defenses are breached, disease development occurs in the patient and it follows a certain sequence. And this tends to be similar for both acute and chronic diseases. So all diseases start with an incubation period. The incubation period, oops, wrong slide. The incubation period is the period from when the patient first is infected with disease and they have their first symptoms. So in between where they have no symptoms but they are infected, that is known as the incubation period. And on the axis here, on the x-axis, is just simply time since your first infection. And then on the y-axis, it's two things. It's the number of microbes growing in the patient, increasing. And it's also the, on the y-axis, it's also the intensity of the signs and symptoms. So the higher up you go, the higher the intensity of the signs and symptoms. So incubation, when you first have disease to the point where you first have signs or symptoms. That is followed by the prodromal period, where the patient has mild signs and symptoms. Like when you're first coming down with a sore throat, you say, geez, am I coming down with a sore throat? You have to think about it before you realize that you have a sore throat. That's the prodromal period, where the patient has very mild signs or symptoms. That ends with the period of illness. Like when you don't have to think about, do I have a sore throat or not? You say, geez, I've got a sore throat. The period of illness is when the signs and symptoms are most severe. It's also often when the number of microbes in the patient are at their greatest. And then the disease breaks and the patient starts feeling better. Like if you have the flu and you have you're sick in bed with a fever, and then the fever breaks. That would be the start of the period of decline, where the signs and symptoms start improving. The number of microorganisms also start decreasing in the patient. The period of decline ends with the period of convalescence. And the period of convalescence is when the patient no longer has signs or symptoms of disease because the number of infective units have gone below a threshold, and so the patient has no signs or symptom. But you notice in the period of convalescence, the patient still has agents that cause disease. Any questions about the stages of disease development? Well,
Can you still get
Go ahead.
can you
there's a
still get other
question.
people sick if you are in the period of convalescence?
Yeah, that was one of my first questions, and that is, what period can you be in that would not cause illness to another patient? Meaning you're not communicable.
None of them, right?
And you were asking the question, are you non-communicable in the period of convalescence? Okay?
You would be communicable in all of them, aren't you?
You are communicable in all of them. You have the infective agent in the incubation period and in the convalescence period, where you have no signs or symptoms in these two periods. And you're communicable in the prodromal. Obviously, you're communicable in the period of illness. And you're also communicable in the period of decline. So you're actually communicable in all of them. Okay? So that's a trick question, and I won't ask you a question like that on the exam. But you are communicable in all of them. And I used to have a mother, and she'd say, oh, I'm getting better. I'm no longer communicable. And I'd say, Mom, you're talking to a microbiologist here. You can't tell me that. You're communicable still. And even after you have the period of convalescence, no more signs and symptoms, for a few days, I'm trying to get my mouse to work. It's not working. For a few days, you are still communicable. Okay? Now, with most adults, for common communicable diseases like the colds and flus, you're actually most infective late in the incubation period when you have no idea you're even sick, and in the prodromal period. These are the two periods when, or late in the incubation period, in the prodromal period, when you're most infective. Why you're not most infective in the period of illness is in an adult, they're mounting an immune response, and because of their immune response, the patients in the period of illness are less communicable than they are in the prodromal period and late in the incubation period. Okay, so that happens with many adults, and my theory is that the adults are trying to prevent the illness from going to their kids, because it would be evolutionarily a bad thing to spread the illness to their children. And so that's my guess as to what's happening. This is not seen in children, okay? They are communicable in every period, and they're most communicable in the period of illness. What happens is the children, they're probably not worried about getting their adults sick, and so they're not mounting an immune response, and so they're most communicable here. I won't ask you to memorize that, but with adults, I'm telling you that you're most contagious for common illnesses like colds and flus late in the incubation period, when you have no signs or symptoms, and in the prodromal period. You're more contagious in those periods than in the period of illness. Now, you are communicable in the period of illness and in the period of decline and the period of convalescence. I'm just saying it's less so than in the prodromal period and then late in the incubation period. All right. Do we always see a prodromal period for a disease?
It's past 2:15.
Oh, is it? Thank you. Let's go ahead and stop here, and I will continue.
I'm going to say no.
Let's continue at 2:32. 2 minutes late. Come on.